Okay, next we're going to talk about how metabolism works and how genetic defects can result in defects in your metabolism, aka, a genetic disease, aka a trait you don't want to have. Well, I've talked about the importance of enzymes. Literally, there are thousands of different types of enzymes in your body conducting most of the chemical reactions. I mean, there are really trillions of chemical reactions that are occurring every second in your body that constitutes living. Digesting food, moving around, sensing the environment, even the cognition of your brain is just a series of chemical reactions. So you were made through development through chemical reactions, and your body is maintained through these chemical reactions. So enzymes mediate all this. These protein structures, they essentially, they work hard to keep you alive. So there are a lot of things we call metabolic pathways, biochemical cascades that are involved in breaking down some substances and making other substances. And one of those that's focused on here is the breakdown of phenylalanine to tyrosine, and tyrosine to hydroxyphenylpyruvic acid. Now, each of these steps is an enzyme-mediated reaction. Requires an enzyme. So it's actually we're going to talk about some more steps here. A step to homogentisic acid, and finally a final step to for let's see if I can pronounce this correctly. Malacetoacetic acid. Whew. Anyway, so a disease is discussed in your book, black urine disease, wherein that if a person has a genetic defect so that they don't produce the enzyme that's necessary for step 4, then the homogentisic acid accumulates and the urine is black. Now, this actually turned out to be not so dangerous, but here you see the entire metabolism or the entire sort of metabolic pathway. And if an enzyme is not produced correctly for any of the steps along this pathway, then that can be a genetic disease. And what happens is that the molecule just ahead of that accumulates. So where do these enzymes come from? Well, the enzymes are made from the instructions in DNA. Now, so a mistake in DNA will lead to an improper enzyme being created, and therefore it's not able to do its job, and a genetic disease will result. And some of these genetic diseases can be kind of bad. Let's take a look at phenylketonuria. Now, this is a disease that causes phenylalanine to accumulate. And when phenylalanine accumulates, it can cause damage to a child's developing nervous system and lead to severe mental retardation. So I'm actually giving this talk while I'm holding a bottle of Dr. Pepper, and I read on the side of the Diet Dr. Pepper, it says phenylketonurics contains phenylalanine. The idea here is that if you have this particular disease, if you avoid phenylalanine in your diet, then you can control the amount that you have. However, if you don't avoid it in your diet, you don't have the ability to break it down, and it accumulates at great levels. So the overall idea here is to sort of show how metabolism happens, how metabolic processes are sort of controlled by enzymes, and that enzymes are made by the instructions in DNA. So if you have mutations in DNA, you're going to have mutations in your enzymes, and you're going to have a genetic disorder. So now let's dig a little bit further into the process wherein the genetic information in DNA is ultimately transcribed into RNA, excuse me, and then ultimately translated into protein structure. Okay, let's zoom in. Now, the first step of the process is transcription, wherein the linear sequence of information in DNA is transcribed into an RNA molecule. RNA uses the same language as DNA, a nucleic acid language, and that's why we call it transcription. It's sort of like just copying, transcribing, like if you were to transcribe a book. However, the next step is called translation, and as you know from literature, if you translate something, you're just not copying it, you're changing the language. So what happens in protein translation is we're translating the information that's in nucleic acid language of DNA and RNA into amino acid language of proteins. And we'll dig into this process in a lot greater detail, but essentially a triplet code is used. There are 3 bases that specify each amino acid. And since there are 4 bases, and you need 3 of them to specify an amino acid, the number of different combinations that you can have is 64, 4 raised to the 3rd power. Okay, now this slide illustrates what we call the central dogma of molecular genetics. You might have heard dogma used before in, like a religious context, but this is the sort of central idea of molecular genetics. That is, the information in DNA is transcribed into RNA. Now, you may be familiar with mRNA or messenger RNA, but there's actually a lot of different types of RNA that are produced, not just the messenger molecule. You have ribosomal RNAs that is produced, and this goes into making the ribosomes that are responsible for protein translation. You have tRNA, and these are the RNA that shuttle amino acids to the ribosome. And there's even other types of RNA like small nuclear RNA that are responsible for splicing messenger RNA, but we'll get to that in just a bit. But anyway, these 3 different types of RNA come together into a structure that we know as the ribosome. We'll be looking at that in a lot greater detail. And that is where protein translation takes place. And the end product is a protein, which then folds up and is further processed to produce, one of the thousands of enzymes, one of the thousands of messenger molecules like hormones or receptor molecules or gates or structural molecules or, actin fibers. The things that essentially comprise a good bit of the structure and the function of your body and the body of organisms on Earth. So let's take a closer look at transcription. Just as DNA can be replicated by pulling it apart and copying both strands, RNA can be made by pulling the DNA molecule apart and then copying one of the strands as RNA. And it's obvious to me that these processes must have evolved from the other, and we'll talk a little bit about that when we talk about the origin of life. Okay, so let's look at protein translation. The idea here is that the message that was originally in DNA is brought out of the nucleus and into the ribosome by messenger RNA, and then these molecules that are also RNA but are RNA that's folded up in different ways to form transfer RNA, they shuttle a series of amino acids to the ribosome, and so and they also have 3 letters that kind of fit together with the 3 letters in RNA. Each of these 3 letters is called a codon, and the 3 letters in the tRNA is called an anticodon. We'll look at that in a lot greater detail a little bit further in the course. But the idea is that through this process, the information in DNA and then RNA is then translated into a sequence of information of amino acids which comprise proteins. Okay, now let's look to see how mutations in DNA can actually cause this genetic disease, phenylketonuria. All right, so here's how the process normally works, is that we've got a gene, PAH gene, P-A-H, that is transcribed and then translated into a polypeptide um, that is necessary for the correct function, the enzymes, that break down phenylalanine. So now if you have a mutation, let's say there's a mutation in DNA, so that this base pair at the beginning, which was AT, is now GC. So maybe there's a mutation in the A, changing it to a G, and so its complement, when copied, is now a C. And so this is, as you might imagine, a bit of a problem. It changes the RNA. That mutation is kind of passed on to the RNA, and then it's passed on to the protein. Now, in this case, it's a particularly bad problem because AUG is the universal start codon. That's the section on mRNA that is recognized, and that's where translation starts. Okay, now let's take a look at another mutation that can occur. You can have a mutation of a C to a T, so the mutation of the complementary molecule in the DNA goes from a G to an A, and let's zoom out just a little bit here. So this single mutation doesn't result in such a severe change in the polypeptide, in the protein, the enzyme down the line, as the last one, wherein there's no polypeptide that's produced because you don't have a start codon. What happens here is that you've just got a single base change results in a single amino acid change. And this slightly different protein has no enzymatic activity. So even though you're producing, a protein that's kind of similar to the right one, it still doesn't work. So it's basically, the same effect on the phenotype, or the same effect on the whole organism, as not even producing the protein at all, which is what the mutation, the first mutation does that we looked at.